Hey guys, my name is Jadon, and let's talk about the ESS 2.3 HL content. Now, for those who are watching this video, this is just a brief overview of the HL content. We are not going in depth into anything. It's just a review. Yes, so let's start by talking about the table of contents. We'll be talking about the methane, nitrogen cycle, human impacts on the nitrogen cycle, the Haber process, and the planetary boundaries. So let's talk about methane. Um, Methane is produced in a anaerobic environment by methanogenic bacteria. Anaerobic basically means oxygen-free place. So some examples would be a digestive system of animals, wetlands, waterlogged soils, yeah, etc. And the residence time of methane is around 10 years in the atmosphere, and it is eventually oxidized to carbon dioxide after those 10 years. And yeah, during those 10 years, methane is a much more potent greenhouse gas than CO2. Right, so let's talk about the nitrogen cycle now. The nitrogen cycle contains organic and inorganic stores. And basically, what they are, well, let's talk about organic. Organic stores are proteins, amino acids, and DNA/RNA that are living in organisms. And inorganic stores are basically atmospheric nitrogen, nitrates, nitrites, and ammonium. Uh, Flows in the nitrogen cycle include mineral uptake by producers, photosynthesis, consumption, excretion, death, decomposition, and aminification. And also, plants can't fix nitrogens directly from uh, the atmosphere, so uh, they rely on mutualistic associations with nitrogen fixing bacteria. So let's talk about the um, nitrogen cycle. So the way that atmospheric nitrogen is turned into ammonium is basically through nitrogen fixing bacteria that live in roots of plants, or it could be through rainwater, or it could be through lightning as well. And basically, nitrogen fixation is the conversion of atmospheric nitrogen to ammonium. And another key definition and keyword that we need to know is nitrification, which is the conversion of ammonia to nitrates. Uh, Denitrification is the conversion of nitrates back into nitrogen, and it usually requires a anaerobic place to do that. Decomposition is the conversion of proteins or amino acids into ammonium. And bacteria have essential roles in these nitrogen cycles, and without them, this cycle wouldn't exist. As you can see in the nitrogen fixation process, nitrogen fixing bacteria is essential, and in the nitrification process, turning ammonium into uh, nitrates and nitrites, these nitrifying bacteria are a essential part of this process as well. So, how have humans disrupted this system, this nitrogen cycle system? So, first, we did it through deforestation, where deforestation leads to fewer trees, which leads to less leaf litter and plant matter. Um, this also leads to less decomposing material and fewer animals, which lead to less nitrogen rich waste such as urine and feces. The second way is through agriculture, mainly through the tilling of the soil and harvesting crops before they decompose, which removes nitrogen from the system, from the crops. And we've also added way too much fertilizers, and these excess nitrates and nitrites usually run off into waterways, causing eutrophication. Eutrophication is basically um, the process, no, not the process, the state of which the water has way too much nutrients, and in this case, way too much nitrogen. This causes dead zones and algal blooms, which are both very deadly for marine life. The third way that we have affected the nitrogen cycle is through aquaculture. We have created a lot of fish farms which produce tons of ammonia, which are then converted by these bacteria into nitrates and nitrites. And high levels of nitrogen, nitrates, and nitrites are toxic to fish. Lastly, we have impacted the nitrogen cycle through urbanization as well. The fertilizers used on lawns, parks, and the movement of nitrogen rich soils cause high nitrogen leaching, which is basically same thing as eutrophication, the runoff of these nitrogens into um, bodies of water. And our pet waste, human sewage, landfills have also added a lot of um, nitrogen into our ecosystem. So let's talk about the Haber process. The Haber process is an industrial process that produces ammonia from nitrogen and hydrogen, and it is used as a fertilizer. Um, the pros of the Haber process is that it increases crop yields, so we don't have to wait 6 months every time to wait for the soil to become fertile for us to plant. And it supports global food production, obviously, and reduces land needed for farming. However, the cons of the Haber process is that it is really energy intensive. There's excess fertilizers that lead to eutrophication, and it produces greenhouse gases as well. So basically, the big picture is that the Haber process has enabled for a green revolution, but um, massively increased global nitrogen pollution as well. Let's talk about the planetary boundary models. Um, so basically, through what we've read in our cognitive assignment, the nitrogen cycle planetary boundary is severely exceeded. Um, According to scientists, our safe limit is about 35 million tons of nitrogen per year, and we are currently using 121 million tons per year, which is 3.5 times more. Um, Again, some of these uh, impacts and consequences of having too much reactive nitrogen leads to water pollution, algal blooms, soil acidification, and all in all biodiversity loss. Yeah. So global collaboration is needed to address this uncontrolled use of nitrogen in industrial and agricultural processes. And yeah, it's essential to bring back the nitrogen cycle back within the planetary boundary models. And as you can see, from 2009 to 2023, it's not getting better. We're just been crossing more boundaries. So yeah, that would be it for what I have to say about this HL content. And thank you for your time.